international scientists each week till mid of July. Today we have a very outstanding researcher, Dr. Ikramy Khalil, who will deliver his research to us through MGBS. And let me please, in a few words, introduce Dr. Ikramy. Dr. Ikramy got his PSC of Pharmaceutical Sciences from Faculty of Pharmacy, Asyut University in Egypt, and then he got a fellowship to Japan, and he studied master and PhD from he got his master and PhD from Graduate School of Pharmaceutical Sciences, Hokkaido University in Japan, and the topic of his master and PhD was in molecular design of pharmaceutics, drug and gene delivery. Dr. Ikramy got his some administrative jobs. He is head of grad committee of clinical pharmacy program in Faculty of Pharmacy, Asyut University, and he has also some other administrative jobs. He is the coordinator of Department of Pharmaceutics and Pharmaceutical Technology at the Faculty of Pharmacy, Tiba University, KSA. He is a manager of the Laboratory of Innovative Nanomedicine in Hokkaido University in Japan. Dr. Ikramy has 32 articles and 8 book chapters. He has H index of 21 and citation of 3,535. Dr. Ikramy, he is a reviewer in high international journals like Journal of Controlled Relays, International Journal of Pharmaceutics, and Biomaterial. Dr. Ikramy, thank you so much for joining us. It's really, we are so happy to have you in the MGBS, and please go ahead for your presentation.
Okay. Thank you, Dr. Ahmed, for your nice introduction. Hello, everyone. It's my pleasure to be with you for second time in MGBS. I was with you in, as far as I remember, 2019 in November, and this is my second time, and it's really my great honor to be with you in molecular genetics and biotechnology seminars in 2022. So first, I would like to thank the organizers, especially Dr. Salam, for accepting my proposal to present with you today. As Dr. Ahmed said, my name is Ikramy Khalil, and I am Professor of Pharmaceutics in Faculty of Pharmacy, Asyut University. And the talk of my today speech is spleen-targeted gene delivery and its implication for cancer immunization. So first, this work, I have to mention, this work is affiliated Laboratory of Innovative Nanomedicine, Faculty of Pharmaceutical Sciences, Hokkaido University, Japan, in collaboration with Department of Pharmaceutics, Faculty of Pharmacy, Asyut University, Egypt. So let me start my talk with a brief introduction about gene therapy or gene delivery. It's not new term now. Everyone may be aware of what is the meaning of gene therapy, but let me just briefly introduce what is gene therapy. As you all know, we still have many incurable diseases, and we need some innovative strategies to find cures for these diseases. And one of these innovative strategies is to use what we call gene therapy. So gene therapy is simply means the introduction of nucleic acids, either DNA or RNA, into a patient to treat a disease. So simply we want to use DNA or RNA as a drug. So if we compare the conventional drugs with the gene therapy, we will find that the conventional drugs are chemicals with small molecular weight. But when we talk about gene therapy, the drug here is large molecular compound like DNA or RNA. These molecules must be internalized into the cell and must be expressed to produce a specific function. So if we talk about gene therapy in the sense of how difficult it is, yes, it's very difficult to internalize such large molecules like DNA or RNA into cells. Why this is difficult? First, these molecules are unstable in vivo if they are administered systemically, and they have very low membrane permeability, so they cannot penetrate the plasma membrane easily. Also, they have inefficient intracellular trafficking. They have many barriers inside the cell, so they cannot go to their sites easily. And finally, they produce some immune response if they are injected in the circulation. Therefore, we have to find some innovative designs to deliver the therapeutic nucleic acids to their target sites inside the cell. So actually, there are two strategies for gene therapy. The first one is gene augmentation therapy, and the second one is gene inhibition therapy. So simply, augmentation therapy, we have some cells with non-functional gene or with some absent genes. In this case, we introduce the functioning DNA, and if it's internalized into the cell successfully, the cell will function normally. And in case of gene inhibition therapy, we have some cells which have some faulty gene or some increased amount of some genes. This will cause the disease state. So in this case, we need to introduce some inhibitory nucleic acids so that it can inhibit the genes and return the cell to function normally. So in the first strategy, the gene augmentation therapy, we can use either DNA or messenger RNA. We all know that DNA, when reached to the nucleus, it will be transcribed into messenger RNA, and then messenger RNA will go to the cytosol, and in the cytosol, it will be translated into protein, which will produce its function. So in the gene augmentation therapy, we can use either DNA or mRNA to produce a certain protein inside the cell. And simply, in case of DNA, we usually add the therapeutic gene as a part of the plasmid DNA vector obtained from bacterial DNA. We introduce the therapeutic gene in the bacterial plasmid DNA vector, and we multiply this gene in bacteria, and then we can use it as a source of therapeutic gene. In case of gene inhibitory therapy, we can use several nucleic acids, but the most famous one is small interfering RNA or siRNA. These molecules can inhibit the translation of the messenger RNA into protein. Therefore, they can produce gene inhibition effect. So in both cases, we can use plasmid DNA or messenger RNA or siRNA as drugs. And if we look at how these molecules work, we should recognize that first, they have to be internalized into the cell because the target site is usually inside the cell, not outside the cell. So we have to pass the plasma membrane. We must reach to the cytosol. In case of DNA, we have to reach to the nucleus. And in the nucleus, the DNA will be transcribed into messenger RNA, and the messenger RNA will go to the cytosol, where it will be translated into protein that can produce a certain function. So also, as an alternative, recently we can use also therapeutic messenger RNA. In this case, the therapeutic messenger RNA doesn't need to go to the nucleus, but it just needs to be delivered to the cytosol. And in the cytosol, it will be translated into protein. So in both cases, we need the therapeutic DNA or the therapeutic messenger RNA to enter into the cell. And as I said, this internalization step is not an easy step. Why? Because these high molecular weight drugs or nucleic acids, either DNA or RNA, they are highly hydrophilic, they have high negative charge, and they have high molecular weight. And these three reasons make it very difficult for them to internalize into the cell. And that's why we usually use vectors, and these vectors will help the DNA or RNA to enter into the cell and to reach their target sites into the cell. And these vectors, we can usually divide them to viral and non-viral gene vectors. And let me explain briefly what is meant by viral vectors. Number one, viral vectors means actually a virus which we remove the viral genome and replace it with the therapeutic gene. So we convert the virus into a viral vector. It has no ability to replicate or to make pathogenesis to the patient. However, it has a some therapeutic gene inside it, so it will deliver the therapeutic gene to inside the nucleus, and therefore we can get the benefit we need. That is a viral vector. In case of non-viral vectors, we have many methods, either physical or chemical. But the most commonly used methods now are chemical methods. For example, we can use what's called cationic lipoplexes. These lipoplexes just a complex between cationic liposomes and DNA parts. And this complex can easily pass the plasma membrane and can deliver the DNA to inside the cell. This is called cationic lipoplexes. And more recently, a new type of lipid is introduced, which is called ionizable lipid nanoparticles. In this case, DNA is encapsulated in pH-sensitive lipids. These lipids will go into the cell. It will make fusion with the endosomal membrane and will release the DNA into the cytosol. So in both cases, now these nanoparticles or lipid nanoparticles are commonly used for delivering DNA into the cell, and this is one of the non-viral vectors used. And now we want to increase the efficiency of these non-viral vectors to compete with the viral vectors, which are more efficient still until now. So now it's actually, we have so many approved gene therapy products in the world. The first product was approved in China in 2003. It is called Gendicine. This is adenovirus vector carrying a specific tumor suppressor gene, P53 gene. And this drug is injected directly into the tumor. It's used for treatment of head and neck squamous cell carcinoma. And as we see, this is the first approved gene therapy product in the world in 2003. And in Europe, another gene therapy product was approved in 2012 called Glebra. And this is also a viral vector, but this type is adeno-associated viral vector carrying the lipoprotein lipase gene. And it reverses the lipoprotein lipase deficiency, one of the genetic diseases. And another one was the first, let me say, the first in vivo gene therapy product approved in USA. It was called Luxturna in 2017, and this is the first in vivo approved drug by FDA. It's used for treatment of hereditary retinal dystrophy, some sort of genetic blindness. It's also adeno-associated virus carrying the gene which is missing in these diseases. This gene is called RBE65. So these three products, if we look at these three products, we find that they are used all locally. Here it's injected into the tumor, here it is injected into the muscle, and here it is localized or added locally to the eye. And also the three share that they are viral vectors. Here it is adenovirus, this is adeno-associated virus, and this is adeno-associated virus. So they all are local and they all are viral vectors. So then in 2017, one product was approved by the FDA called Kymria. This Kymria is the first gene therapy product approved in USA, not in vivo. It's called ex vivo because it is actually composed of genetically modified T cells. We call it CAR T cell therapy, and it is used for treatment of some kind of leukemia, B-cell acute lymphoblastic leukemia. And actually the recovery rate is very high, almost 82% complete recovery from this type, from this difficult type of cancer. And as we see, if we look at this Kymria, we find also another product approved in the same year called Yescarta. It is almost the same technology, and both are used ex vivo, which means that they are cells which are modified in vitro, then re-infused back to the patient. And both are still viral system because these cells are transfected with viral vectors. So the first non-viral therapy approved for gene therapy, it's called patisiran. This is in 2018. This system is siRNA molecule, and it is composed of nucleic acid lipid nanoparticle. As I said before, this is one of the non-viral vectors and encapsulating siRNA, which means this is gene inhibitory therapy. And this drug was approved for treatment of hereditary transthyretin amyloidosis, also one of the genetic diseases, and it targets the liver hepatocytes after intravenous administration. Is a type, as I said, type of siRNA lipid nanoparticles. And also, as we see, this patisiran is very important because it is the first in vivo and systemically administered system. It is the first non-viral approved product. It is the first RNA-based approved product. So this drug, the approval of this drug, actually raised our hopes that we will have in the future much more products which can be used for treatment of many diseases which cannot be treated now. And actually in 2019, another product was approved for treatment of some type of child spinal muscular atrophy. And this is also still viral vector because it's adeno-associated virus carrying the missing gene, in this case SMN1 gene, and used as intravenous infusion. So as I said, we still have we are still waiting for more future-based therapeutics. And I believe that in the coming few years, we are going to hear more and more drugs approved by the FDA. The price of these drugs now are very high, but I'm expecting that it will go down by years because it will be, I think it will be normally used in the future, and it will solve many problems which we cannot solve until now. So that's just a simple idea about gene therapy. And let me talk about reality that now we are using gene therapy actually in COVID-19 vaccines. If we are following, there are two types of vaccines available now, the conventional vaccines and the nucleic acid vaccine. Nucleic acid vaccine is actually a type of gene therapy product. The conventional vaccine, as we know, like Sinopharm or Sinovac, these kind of live attenuated virus. It is the COVID-19 virus, but it's live attenuated. It has no ability to replicate. So it just stimulates the immune system, but it cannot make any infection or pathogenesis to the patient. And these conventional vaccines, as I said, we use the same virus, but live attenuated. However, in the more new nucleic acid vaccines or gene therapy vaccines, we can use DNA as a vaccine, or we can use messenger RNA as a vaccine. In case of DNA, we include the gene specific for the spike protein of the virus. We include it in a plasmid DNA vector, and this DNA can be incorporated into a viral vector to be easily internalized into the cell. And in case of messenger RNA vaccine, we also have the messenger RNA part, which encodes the spike protein of the virus, and this is enclosed in some type of lipid nanoparticles, and this can be injected intramuscularly. So these nucleic acid vaccines, as I said, for example, the DNA vaccine, Johnson & Johnson and Oxford AstraZeneca vaccine, both are DNA vaccine. They are viral vectors, while the Pfizer or BioNTech and the Moderna vaccine, both are messenger RNA vaccine. So as we see, now it's reality. Now we are using gene therapy products actually in our life, and it's one of the solutions that offered for us to fight some difficult situation like the COVID-19 pandemic. So still in the nucleic acid vaccine, if we look at what happens if we inject for example, the viral vector or the messenger RNA lipid nanoparticle. The viral vector will deliver the DNA into the cell, and this DNA will be translated into the nucleus into messenger RNA. This messenger RNA will finally be converted into the spike protein. This spike protein will be taken up by some antigen-presenting cells like dendritic cells or macrophages, and they will digest these spike proteins and will produce the peptide fragment on the spike protein on the surface of this cell antigen-presenting cells, and this will produce immune response against this spike protein of the virus. In the same way, when we talk about messenger RNA vaccine, as I said, this is lipid nanoparticles encapsulating the messenger RNA. When the messenger RNA is delivered to the cell, it will be translated to protein, which is the spike protein, the coronavirus spike protein. This spike protein will be delivered to the antigen-presenting cells, and this here it will be degraded, and some peptide fragment of the spike protein will be presented on the surface of antigen-presenting cells and will produce immune response against this spike protein. So if we look at this system, we find that we need to stimulate the antigen-presenting cells. And here, when we inject these vaccines intramuscularly or subcutaneously, we are transfecting the muscle cells, and also we are transfecting the antigen-presenting cells near the site of administration or near the site of action. However, the antigen-presenting cells are mainly found in lymphoid organs. If we look at the body, so many lymphoid organs. The lymphoid organs, either primary lymphoid organs like the thymus or the bone marrow, or secondary lymphoid organs like the spleen or the lymph nodes. In this case, like for example, spleen and lymph nodes specifically, they have very high number of antigen-presenting cells in very close proximity to the immune cells of the system, like B cells and T cells, which is responsible for producing the immune response. So if we talk about delivery of the genes directly to the spleen or to the lymph nodes, we are expecting that we will have much higher immune response. Therefore, we can use much lower dose compared to the delivery by intramuscular or subcutaneous administration. So if we talk about systemic delivery of lymphoid organs, systemic delivery means we inject the drug mainly in the blood. So if we inject these DNA or RNA or messenger RNA particles into the drug, into the circulation, if we successfully target some lymphoid organs like the spleen or the lymph node, for example, let's say that we are targeting the spleen. If we can target the spleen, if we can transfect some cells in the spleen, which is immune responsive cells like dendritic cells, macrophages, we call it antigen presenting cells, like B cells, which produce antibodies, and like T cells, which is responsible for the cytotoxic T lymphocyte activity. If we successfully make this kind of spleen targeting, we are expecting that we can produce a very strong immune response, which is much higher than the local administration like we have now in COVID-19 vaccine, for example. In this case, we can use much lower dose, and the side effects will be, of course, much lower than the intramuscular or subcutaneous injection. So the aim of this study is to avoid the barriers to the systemic delivery, and we want to target the genes, either DNA or RNA, after systemic administration into the blood. We want to target the spleen specifically. We need to transfect some immune cells in the spleen. So let me summarize the barrier to systemic delivery, because if we inject nucleic acids in the circulation, it will not easily go to the spleen, for example, or to the lymph node. For example, first, the DNA or RNA will be degraded in the plasma. Second, it will have many non-specific interactions, and it will have some binding with serum proteins because they are charged. And finally, it will also taken by the reticuloendothelial system, and it will be degraded by macrophages in the liver and spleen. And also, it is very difficult to pass the vascular system. It has very low vascular permeability, so it will not go out of the blood vessel easily. Finally, it will have no ability to target specific cell population. If we check the destiny of some nucleic acids which injected intravenously, we will find that these large particles will be delivered to the lung endothelium, because this is the first capillary they will be encountered after injection. And also we will find that after some time, 1 hour or 2 hours, the dose will be accumulated mainly in the liver and spleen, but they will be degraded by macrophages, and therefore we almost have no effect. So there are many barriers to the systemic delivery of nucleic acids. If we want to deliver the nucleic acids to the spleen after systemic administration, we have to avoid these barriers. So now the aim of this study was simply to develop a systemically administered non-viral gene delivery system, which can first specifically target the spleen after intravenous administration. Number two, efficiently transfect the immune cells in the spleen, like B cells, macrophages, dendritic cells, and T cells. Number three is to produce immunological response against specific antigens, which we can encode in the plasmid DNA or in messenger RNA. And if we can transfect B cell, for example, we expect to produce antibodies against this antigen. If we can transfect T cells, we can stimulate CD4 T cells or helper T cells to stimulate other type of cells. And also we can stimulate CD8 plus T cells, which we call cytotoxic T lymphocytes. This is responsible for attacking the antigenic viral particles and destroy it. So if we transfect B cell or T cell, we expect to have immunological response. Also, if we can transfect the dendritic cells or macrophages, we can stimulate the activation of B cells and T cells. Because as I said, these cells are called antigen presenting cells. So they will produce the antigen in the form that can easily stimulate B cells and T cells in the spleen or in the circulation. So after developing this system, our aim is also to apply this system for immunization. Actually, our laboratory was not involved in the viral vaccine area of research, but we were mainly concerned with treatment of cancer. So we wanted to apply this system for developing an immunization for cancer. We want to have, if some people, for example, they have high ability to have a certain type of cancer, they can receive an immunization. Like we have immunization against any virus or any disease, we want to have immunization against specific type of cancer for the susceptible people. Therefore, we can protect them from developing cancer. Also, we can use it for also treatment of already existing cancer. So the second aim is to apply this system for immunization and for treatment of cancer. And finally, I wanted to compare between the systemic administration into the blood by intravenous injection and the local administration of the nucleic acids, DNA or RNA, like we are using now in COVID vaccine administration. We are using now intramuscular injection. So which is better, to use intravenous or intramuscular, and which will produce higher immune response? That's also one of the aims of this study. So let me start with the first systems that we started to develop. We developed lipid nanoparticles encapsulating plasmid DNA, and we just coat with plasmid DNA in a lipid system. This lipid system, to be efficient, we decorated the surface of this lipid system with a penetrating peptide, a cell-penetrating peptide called sterylated octoarginine. This positively charged peptide will increase the internalization into the cell and increase the transfection activity. However, the presence of high positive charge on the surface of these nanoparticles make it not suitable for in vivo use or for systemic administration. Therefore, one strategy is to use, we call it a double coating strategy for improved DNA gene expression. So let me explain simply. First, we encapsulate the DNA in a certain lipid containing the R8 or the sterile R8 peptide, and then we cause a second coating step. This second coating step, the main purpose of the second coat is to make more protection for the DNA in the circulation and also to mask the positive charge of the peptide. Therefore, they can be easily administered by in vivo administration. But we all know that if we mask the positive charge, the efficiency of the system will decrease. So our challenge was how to decrease the positive charge so that we can use it systemically in the body. Two we still want to have some high activity. And that's actually after several optimization steps, we could develop this two-step coating system or double coating system for gene expression. And this is the result in HeLa cells. HeLa cells means that's in vitro experiment. And this is a comparison between one-step coating and the two-step coating. And as we see here, when we use the lipid, the main lipid called YSK with the helper lipid called DOPE without sterile R8, the post-step one-step or two-step coating didn't produce high transfection activity in vitro. When we used the helper lipid and the peptide without the main lipid YSK, it also didn't produce high gene expression. However, when we used all three components, the pH sensitive lipid YSK and the helper lipid DOPE and the peptide sterile R8 peptide, when we used the three system, and as we see here, there is a big difference between the first step coating and the two-step coating. So from this study, we conclude that although the second coating decreased the positive charge of the system, but we still have very efficient system that we can transfect HeLa cells efficiently. Next, we go to the systemic administration for this system. We injected these particles intravenously into the circulation, and we measured the gene expression in different organs. So for example, in the liver, both produce high gene expression, but still not so high. Also, the activity in the lung was not so high, and surprisingly, we found that the two-step coating system produced high gene expression in the spleen specifically, and the activity was higher than in the liver and much higher than in the lung. This shows that this system can be particularly efficient in case of transfection of the spleen. Therefore, we wanted to optimize the system more to increase the transfection activity. Actually, the transfection activity here reached a little above 10 to the 5, but we wanted to reach to more than 10 to the 6. So we needed to improve the activity at least 10 times to be applicable for using for in vivo administration. And that's I will go rapidly about the optimization of the system. For example, here we optimized the total lipid amount, and we found that using 320 nanomole of the lipid for 10 microgram DNA is the best ratio. Also, we optimized the amount of the main lipid, the YSK lipid, which is a pH sensitive lipid, and we found that using 40 mole% of the lipid YSK is the best result. And also we optimize the amount of cholesterol in the system. Cholesterol is used to stabilize the system, and as we see here that using 15% of the system as cholesterol produces the optimum effect. And now as we see, we are approaching 10 to the 6, which means that we improved the gene expression efficiency a few times. And also we optimize the amount of BEG or DMG PEG amount. This lipid is responsible for making some lung circulation in the blood, and this also improves the gene expression. And we found that 3 mole% of BEG amount is the optimum ratio that we have used. Now the optimum system produced higher than 10 to the 6 gene expression level, which means that we successfully optimized the system for high gene expression in the spleen. And this is to confirm the synergism between the lipid YSK and the lipid and the peptide, the steryl R8. And as we see in this one-step coating, using YSK is better than the conventional lipid DOTAP, and even with steryl R8 with YSK is better than DOTAP with steryl R8. And also the two-step coating produced much higher activity than the one-step coating. Therefore, this is the optimum system, and we can use it successfully for transfecting spleen cells. So next, we wanted to confirm, can we reach to some immune cells in the spleen, or we just transfect normal parenchymal cells of the spleen? So we started to make the cell suspension from the spleen, and we used specific antibodies to separate the dendritic cells, the macrophages, and the B cells, and the T cells. And we started to check how is the delivery to these cells. We used fluorescence labeled nanoparticles, and we tried to count the fluorescence in case of each cell type. So for example, the number of positive cells in case of T cells was very low, which indicates that we couldn't transfect the T cells in the spleen. However, we can transfect more than 40% of the B cells, about 25% of the dendritic cells, and about 80% of the macrophages. These 3 cells can be used as antigen-presenting cells, which means that we can successfully transfect these 3 cells, and these 3 cells can act as antigen-presenting cells, which can produce immune response, as we see later. So also, we started to check the prophylactic effect against cancer. Here, we used our system as a vaccine against cancer. Let me explain the method, how we use it. For example, at the beginning of the experiment, we injected 30 microgram of the OVA plasmid DNA. This is plasmid DNA which contain a gene specific for antigen against a specific tumor. So if immunization is successful, the body will produce OVA antibody. This antibody will fight the OVA tumor cells. So after 7 days, we inoculate the OVA tumor cells, and if there is antibodies, the tumor will not grow. But if there is no antibodies, the tumor will grow normally. So after the injection or the inoculation of the tumor cells, we followed the tumor growth for about 1 month. And that's the result. As we see here, the tumor volume increased significantly after, in case of control mice treated with PBS buffer or treated with non-specific plasmid DNA. But when we used the specific DNA in our system, this DNA is encoding for the antigen, and therefore it's expected to produce immune response. We found that almost no tumor growth we could observe until 1 month after inoculation of the tumor cells. This indicates that the efficient gene delivery to the spleen with the double-coated system that we developed in this study is a very promising device for vaccine application. It can be applied for example, tumor like in this case. It also can be applied for against infectious diseases like COVID-19, for example, in the future. And also we tested not the prophylactic effect, but we wanted to use the system as a therapy, as a treatment for cancer. Therefore, we first initiate the cancer in the mice, and then we tried to treat it. So first, we inoculated the tumor cells, and after 7 days, we injected the OVA-specific encoding plasmid DNA in our system. We repeated the dose after 10 days and also after 14 days. Then we continued monitoring the tumor growth. And as we see here, the tumor growth in the control mice, PBS or naked plasmid DNA, the tumor growth was much higher than in the case of the treated mice with our system, which indicates that the spleen-targeted system has also the ability to treat existing tumor, not only to produce vaccination or prophylaxis against the cancer. Although in this case, the tumor growth is still increased because it's already existed, but there is significant difference between the control mice and the treated mice. Therefore, we successfully applied our system for the prophylaxis against specific type of cancer and also for treatment of some type of cancer. And next, we wanted to search for more lipid formulation that can produce higher spleen gene delivery. Actually, we started to make a screen to find another lipid which can transfect the spleen more efficiently. In the current study, the previous study, we were using a lipid called YSK05. This is a pH-sensitive lipid, and it showed high activity in combination with the R8 peptide. But we screened different lipid and we compared to find another stronger lipid, and we found one of the less used lipid called DODAP lipid. This lipid is also pH-sensitive lipid, but it's very inefficient in transfection in vitro over cells. So therefore, it's not commonly used. But for our surprise, when we compared the spleen activity after intravenous administration, we found that this system showed high gene expression. For example, this is the result that we have when we compared this YSK05 lipid with the DODAP lipid. And as we see here, the activity become much higher than 10 to the 6. Previously, we just was higher than 10 to the 6 a little, but now we almost 5 times higher than 10 to the X, which means that this DODAP system is more efficient than YSK system in transfecting spleen. And that's all very surprising because, as I said, the DODAP system is less efficient than the YSK system in vitro. We confirmed this result in HeLa cells, and here, as we see, the YSK activity is higher in case of in vitro HeLa cells than the DODAP system, which means that our system is actually not working very good in vitro. However, it works very well in vivo after systemic administration, specifically in the spleen. It also transfects the liver and the lung, but much lower than the spleen, which actually we need. So we started another optimization for the DODAP lipid system, and when we compare DODAP with YSK05, we found that, for example, the delivery to the spleen is much higher. YSK05 is delivered to the spleen much higher than the DODAP lipid system. However, the gene efficiency is higher for DODAP than the YSK. So this means that the DODAP system, although it's not highly delivered to the spleen, but it can produce very high gene expression. The efficiency of transfection seems very high. That makes this system very promising to develop or to update our system with a more stronger system. So here we examine the effect of the helper lipid. Do we need to include helper lipid or not? And for our surprise, when we use, this is the molar ratio of the helper lipid and the main lipid, which is DODAP. And as we see here, when we use the DODAP system without helper lipid, the activity was very low in the liver or the lung or the spleen. However, just by including 10%, 15%, or 20% of the helper lipid, it produces high transfixion activity, specifically in the spleen, not in the liver or the lung, which means that the presence of the helper lipid is essential for spleen transfixion. And when we observed the system in the blood after injection, we found that the system is almost cleared from the blood after 30 minutes, which means that they are clearly rapidly taken up by the liver and by the spleen, and they can have the ability to transfix the spleen cells very efficiently. So here we examine the biodistribution, and here when we used fluorescence labeled system, we used live imaging to check the organs and how much delivery we can do. As we see here, this is a non-treated cell. The organs almost have no fluorescence. But when we use the DODAP LNP system, we can find fluorescence in the liver and spleen and the lung, which is consistent with the previous result. And when we check the gene expression level, also we found non-treated cell produce almost no gene expression, but the DODAP LNP treated cells produce high gene expression, specifically in the spleen, very low gene expression in the lung, and very low and localized gene expression in the liver, which means that this system is specific for spleen transfixion. Then we also confirmed the splenocytes delivery. Again, we deliver mainly to the B cell, dendritic cells, and the macrophage, due to the high number of B cells. So the total fluorescence actually comes from B cells, not from D cells or macrophage, because B cells represent more than 60% of the immune cells in the spleen. That's why the total fluorescence comes mainly from the B cell. Also, we check the gene expression. Can we transfect, can we produce the gene, the protein inside these type of dendritic cells or macrophages or B cells? As we see here, we cannot transfect T cells, but we can successfully transfect B cells, we can transfect dendritic cells, we can transfect macrophages, which indicates that we can make antigen presentation successfully in the spleen. Standardized gene expression showed that the highest gene expression comes from B cell, but also significant gene expression comes from dendritic cells and from macrophages. The lowest gene expression comes from T cell. So we wanted to confirm the B cell involvement in this gene expression. So we used antibody. This antibody can block or can deplete the B cells, and we found that the activity dropped too much, which prove the involvement of B cell receptors in this transfixion process. Also, we wanted to confirm the involvement of complement receptors. These complement receptors are found in the surface of macrophages and B cells, and when we used the antibody against the complement receptors, also the activity strongly dropped due to using this antibody, which means also that the complement system is actively contributing in the transfixion step. Also, again, we checked the prophylactic effect successfully, and also we checked the therapeutic effect, and also successfully we could treat the already existing tumor. And then we actually wanted to evaluate the immune response. First, we wanted to evaluate the CTL activity, which means cytotoxic T lymphocytes activity. Let me explain rapidly. If we successfully stimulate T cells, we will stimulate CD8 T cells. These CD8 T cells will be transformed to cytotoxic T lymphocytes, which can attack the cancer cells and kill it. We here measured the killing percent, and as we see here, the control didn't show any killing of tumor cell. And the system, when we used non-specific plasmid DNA, this is like a control, also didn't show high specific killing of tumor cells. But when we used our system, we almost have about 80% of cytotoxic T lymphocytes. The killing of the OVA-specific tumor cells was about 80% of the total cells, which indicate very high specific cytotoxic T lymphocyte activity. Again, this activity was dropped when we block the B cell by using antibody. This means that this cytotoxic T lymphocytes is actually mediated by B cell. B cell act as antigen presenting cell, which will introduce the antigen into the T cell, and then T cell will be activated to produce cytotoxic T lymphocyte activity. Also, we checked the production of antibody, and we found that also if we transfect B cells, we can produce antibody. Here, this control didn't show specific antibody production. Also, the non-specific DNA used as a control also didn't produce high antibody. But when we used our system, we can produce at least four-fold amount of higher antibody against the tumor. Therefore, we can say that our immune response comes from both humoral immunity due to antibody production and cytotoxic T, our cellular immunity due to the cytotoxic T lymphocyte activity. Also, when we blocked the amount, the B cells, we significantly lost the activity or lost the antibody production, which indicates that B cell play a major role in this process. So I'm very sorry if I'm taking long time, so I have to jump to the next system. And as I said, in the previous system, we are using DNA. And actually, as I said in the introduction, we can use messenger RNA, not plasmid DNA. Messenger RNA is much more efficient. Therefore, we started to use the same system, but instead of using plasmid DNA, we used messenger RNA. Messenger RNA doesn't need to reach the nucleus. It just needs to reach the cytosol. Therefore, it's very efficient compared to the therapeutic DNA. Although it is less stable, it is much more expensive, but it's much more efficient. Therefore, we started to test our system for messenger RNA delivery. As we see here, we prepared the system with two types of method, one called hydration method and one called dilution method, and both methods produced high gene expression over 10 to the 6 in the spleen. This is used with very low dose, much lower than the previous. Therefore, we expect that if we increase the dose, we can reduce much more activity. Here, when we check the effect of the helper lipid, DOPE, we didn't find a role of the helper lipid in the spleen, although the presence of the helper lipid decreases the activity in the liver, which means increase the specificity and reduce the side effects. So therefore, we continue using the helper lipid in the next experiment. Here, when we increase the dose, the activity in the spleen is increased, and now we reach it over 10 to power 8, which means very high transfection activity in the spleen. That's actually as comparable as the viral vector, which means that this system is very efficient for transfecting the spleen cells. And also we continued the same. We confirmed the spleen delivery. We also checked the gene expression in the spleen, and again we successfully transfected dendritic cells, macrophages, and B cells, but we cannot transfect T cells. But these antigen presenting cells can actually indirectly stimulate T cells to produce CTL activity. Also, we tested the prophylactic anti-cancer effect, and again, the mice prophylactic or the mice received the vaccine almost didn't show any tumor growth, although the control showed significant tumor growth in all other control mice. Also, therapeutically, we can have significant effect comparing the control mice and the treated mice. So it can be used as both prophylactic and as treatment. Even the immune response, we could compare the CTL activity as we see here. We used different doses of the messenger RNA, and even very low dose like 0.05 milligram per kilogram. This is very, very low dose compared to the normal dose. Normal dose is 1 to 10 milligram per kilogram. And as we see here, even low dose can produce high specific killing to the tumor cell, and even as low as 0.8 milligram per kilogram produced almost 100% OVA specific killing of the tumor cell. So the CTL activity is very, very high after the injection of the messenger RNA, which targets the spleen. And here, unfortunately, the antibody production was not produced in the same manner as the plasmid DNA system, which shows that the messenger RNA, the activity comes mainly from the cytotoxic or cellular immunity and not from the humoral or antibody immunity. That's a big difference between the messenger RNA and plasmid DNA, and I think it needs more investigation to understand the mechanism why the system is different in the mechanism of the immune response. And also, we finally wanted to compare with a system that we wanted to test our formulation, how strong we are. So we searched it until we found a paper in Nature which produced a specific gene expression in the spleen, same as we do, but they used a system, lipoplex system, just control cationic liposome complexed with messenger RNA. So we compared our system, the DODAP system, with the lipoplex system. When we compared the gene expression, the lipoplex system published in Nature was producing much higher gene expression than our system in the spleen. However, when we used the immune response, we found that our system using two dose, high dose and low dose, produced almost higher killing of tumor cells than the lipoplex or the Nature lipoplex system. Therefore, our system can compete even the most strong system which is published so far regarding producing anti-tumor activity after vaccine receipt. And finally, I compared the systemic and local administration. Here I used our system intravenously and intramuscularly in the same dose. This is low dose, so we can produce about 60% using intravenous, but less than 25% when used intramuscularly. This is clearly shows that the intravenous administration is much more efficient pathway to inject these systems, much higher than the intramuscular injection. Also the same result we have, but lower killing efficiency when we use the standard RNA lipoplex system. Also the intravenous was higher than the intramuscular. Therefore, this is very important result, because in the future we may shift the vaccine which we deliver now by intramuscular injection. In the future, I believe we will go to the intravenous route, and we will use much lower dose than we normally use now. So the side effects will be much lower than what we expect now. So finally, as a conclusion, I showed in this presentation that we have developed 3 systemically administered systems. They are all non-viral gene delivery systems that can be transfecting spleen cells very efficiently. One called YSK05 nanoparticles for plasmid DNA, and one called DODAP nanoparticles for plasmid DNA. And this same system also can be used for messenger RNA delivery. Of course, this is the highest activity we could produce by using the messenger RNA delivery. All these 3 systems can specifically target the spleen after systemic administration. They can efficiently transfect specific immune cells in the spleen, namely the B cell, the dendritic cells, and the macrophage, and they can efficiently produce immunological response. In the case of plasmid DNA system, we can produce both cellular and humoral immunity. However, in case of DODAP nanoparticle messenger RNA, we can only produce cellular immunity, although the activity is almost similar or higher in total. And finally, these 3 systems can all be applied for cancer immunization. They successfully transfect cancer and prevent cancer development, and also they can treat the already existing cancer. And finally, I proved that the systemic delivery of the nucleic acid is a very promising route of administration compared to the currently used intramuscular or subcutaneous injection. So finally, the research I showed today is published in Journal of Controlled Disease in 2019, and also in the same journal in another issue in 2021. This journal has impact factor 9.776. This is the latest one. And let me acknowledge Mr. Seigu Kimura. Seigu Kimura, he was a student during the preparation of these papers. He was master course student. Now he is PhD student and still working in Hokkaido University. And also I would like to acknowledge Mr. Ryoyo Shimusakai. Shimusakai is a master course student now in Hokkaido University, and he is now developing the messenger RNA nanoparticles that I showed in the last part of my presentation. So again, I would like to acknowledge that this research is supported by special education and research expenses from the Ministry of Education, Culture, Sports, Science and Technology Mixed of Japan. And I would like to acknowledge all the members of the Hokkaido University Graduate School of Pharmaceutical Sciences, especially my professor, my supervisor, and the head of the department, Professor Hideyoshi Harashima. So thank you for your listening, and I'm very sorry for my long talk, and I'm ready to receive questions.
Thank you, Dr. Thank you, Dr. Ikramy, for this interesting lecture. Actually, it has a lot of massive information about gene therapy. And now we open the time for discussion. Dr. Ikramy, I think we have some questions in the chat room. Can you read it, or shall I read it?
I will go to the chat.
Yeah, chat room from Gamal Yassin.
This one?
Yeah, the first one was, what about the effective dose? This is because he has some question also.
So the effective dose. Okay, so let me say, in case of DNA, we use about 30 microgram per mice, which is normally lower than the used in other papers, but we can produce the same response almost. But however, when we use messenger RNA, let me say, for example, that the lipoplex system that I showed in Nature, they used the dose is about 10 microgram, but I showed the result here today with 0.8 microgram, which means that about 10 times lower than the dose that we use. So actually our system is very efficient in transfecting spleen cells, even when we use much lower dose than normal. So now we can transfect even using less than 0.01 milligram per kilogram messenger RNA, which is very low dose, almost has no side effects and almost much lower than the normal dose that we inject now intramuscularly, for example. So is it answer your question or still? I'm sorry, I can't see the questions, Dr. Ahmed.
No, yeah, there is like another question. What type of increased expressed genes in spleen and other organs?
Okay, so we use two types of experiment. First, when we make optimization of the system, we call it marker gene, which is a certain gene that we can measure. It's not therapeutic, but just something we can measure, so we can evaluate the efficiency of our system. So we use luciferase gene. This luciferase gene produces some substrate. When we add to a system, there is an interaction happen, and we can measure the light produced from this interaction. So this is during the optimization. So when we optimize, I measure the light intensity or something we can measure. But when we go to the anti-tumor response, we are using specific DNA against the tumor. This DNA, if reached to the cell, it will produce the tumor antigen. So the tumor antigen, if produced in the cell, it will make antibody, it will make cellular immunity, and therefore we use a specific plasmid DNA. For example, for the tumor cell, we use OVA-specific plasmid DNA. This plasmid DNA is encoding a certain gene against a certain antigen against the OVA tumor cells. So that's also one. And of course, when we measure the antibody or the immune response, we have to use also the specific antigen encoding plasmid DNA. So as I said here, we are using two types of plasmid DNA or messenger RNA. One for optimization, in which we use non-specific, just something we can measure. And the other one, in case of tumor study or in case of immune response measurement, we use a specific plasmid DNA or messenger RNA, which encodes the antigen against the tumor. Like in our case, in case of COVID-19, for example, they are used DNA or messenger RNA against the spike protein of the virus. So if these molecules go into the cell, they will produce the spike protein of the virus in the circulation, so the immune system is stimulated. The same we do here, but instead of the spike protein of the virus, we use antigen from tumor cells. So we produce antibody and cytotoxic T cell lymphocyte activities against the tumor cells. So when we inoculate the tumor cells, there is a strong immune reaction, and the tumor killing is very high. As I showed, it reached almost 100% of tumor killing percent.
Okay, for anyone, just if you would like to speak with the mics, just you can raise your hand, and just I will allow you to tell your questions. We have another question, Dr. Ikramy. Can we use this drug type side by side with the other treatments, chemicals, or the others?
Yes, of course we can use, but simply this kind of therapy should solve the problem from its origin. So like when we talk about some genetic disease, if you can correct the genetic cause of the disease, you don't need another drugs. All drugs we are using now is just symptomatic, just treat the symptoms, but not treat the cause. But if we use gene therapy, we can treat the cause. So I believe we don't need to use any other conventional therapy. Maybe at the beginning, until the system is adapted to the body, maybe at the beginning we use some supplementary method. But once the gene therapy system is already in action, it doesn't need any additional treatment, in my opinion.
Okay. For how long can this dose to keep effect?
That's very important question and very critical. Actually, so far the prophylaxis effect against the tumor, just one dose is enough, as you see here. After we inoculated the tumor cells, we followed for about 1 month, and we couldn't find any sign of tumor, which means that the antibody production is very strong, and even 1 month after tumor inoculation, we can't find any tumor. So that's in case of prophylaxis study. But in case of anti-tumor study, in case of already existing tumor, we needed to inject 3 doses at day 7, day 10, and day 14. Actually, we don't know exactly if we need more in the future, but at least we can have significant anti-tumor effect by using these 3 doses. And I think we need at least 1 dose, but the second and 3 dose may be supplementing. But I believe these 3 doses is enough to produce significant amount of antibody that maybe we don't need to add more doses in the future. But actually, we didn't test more than 3 doses until now.
Okay, thanks, doctor. Can we apply this type of for more than one cancer type?
Yes, of course, we can make plasmid DNA. Now we are using plasmid DNA encoding only one antigen against specific tumor cells, but we can make any kind of antigen we can include in the plasmid DNA. So we can have plasmid DNA which have 2 or 3 antigen or even more, but yeah, theoretically it's possible, I think.
Okay, thank you, Dr. Ikramy. I have my question.
Yes.
My question is, can we use such systematic delivery for treating more than genetic disease? I mean,
Uh, yes, please continue. Oh, okay. Can you repeat the question again, Dr. Ahmed?
okay, can we use such systematic delivery for treating more than one genetic disease like diabetic and cancer and
Yeah, yeah. In case of diabetic, it's still difficult because you have to deliver the gene to the pancreas or pancreatic cells, which is not easy to pass the vasculature and go to the pancreas. But in case of tumor cells, it's actually possible because not only we can make vaccination or transfect spleen cells, we have some studies how to target the tumor cells. So we have many systems that we can use to target tumor cells in the body. So yes, it's very possible. It's actually, I think, more than 60 to 70% of the clinical trials of the gene therapy products are all for treatment of cancer.
of
So
course. So looks like this method is specific for tumor cancer or
no, it is just because it is easy to deliver the
oh,
system to tumor cells,
okay.
much easier than other normal tissue.
Ah, okay.
So, and let me explain again, because in case of tumor, the vasculature is not perfect. So the capillaries have many spaces. So our system can escape and can go to the tumor. And then on the side, on the surface of tumor cells, we have many receptors that we can use to target our system and can make it highly delivered to the tumor cells. But in other organs like pancreas or lung, it's very difficult to go out of the circulation because the capillary is continuous and is perfect capillary, no spaces. And also, we don't have so many specific targets for specific type of cells like in case of tumor. Tumor, there are many receptors overexpressed in tumor cells, so we can use it as a target. But in other cells, we cannot escape from the circulation first. Second, we cannot have so many receptors to target other kind of cells. But we can transfect, for example, we have some studies about targeting the lung endothelial system. Lung endothelial cells is the cells which um, uh, which found in the vasculature in the lung. In this case, we can target the lung vasculature. We can prevent the growth of the lung vasculature, which means we can suppress the tumor growth in the lung. Also, we have some studies about this kind of system. We also have some studies to target the liver hepatocytes to prevent liver cancer. Also, we have some kind of specific receptors that we can use to target hepatic cells. So number one, easier target is the tumor cells, and the second easier target is the liver cells, hepatocytes, and the third one is the lung endothelial cells or the lung capillaries specifically. And of course, the spleen cells we can now we can target, as I showed in my presentation, using some specific lipids.
Okay, thank you, Dr. Ikramy. Do we have other questions from anybody? Just if you'd like to speak or to tell your question, just raise your hand, and you can tell your questions or discussion as you need. Okay, I think we don't have further questions. Again, thank you, Dr. Ikramy, for this very interesting, really, lectures, and you answered all questions very clearly. Thank you, Dr. Ikramy. Thank you again.
Thank you, Dr. Ahmed, and I'm very grateful for let me being a part of these seminars. And I would like to thank all the attendants. Thank you very much for your interest in my talk. And I think if you have any question in the future, you please feel free to contact me. I think I have my email on my page on the university. So please feel free to send me any query about my lecture or any other type of question, and I will respond, inshallah, to your email, inshallah.
Okay, and the email
Thank
of
you,
Dr.
Dr.
Ikramy, the email of Dr. Ikramy, you will find it in the poster that you received in the email
I
from
can
us.
write it now.
Yes, it will be nice. You can write it in the chat room, please, Dr. Ikramy.
I wrote it in the chat. Did it appear? I can see it.
Okay, please register for the attendance in the link in the Google form. So in case of you would like to receive a certificate of attendance. As you know from our email that you will receive a certificate of attendance, but this will be at the end of our seminars. We cannot issue a certificate for each seminar due to time load. So therefore, before ending our seminars, we will send a specific email and specific information how to get the certificate of attendance. Thank you so much for all attendees to our lectures, and see you next week. So, goodbye.
Bye.
We can also keep the link for 15 minutes more if anyone would like to register his email address and the name.